Our next speaker is Abby Johnson. Dr. Johnson is Assistant Professor in the Division of Epidemiology and Community Health in the School of Public Health at the University of Minnesota. She is also Associate Director of the Nutrition Coordinating Center, which distributes and supports the Nutrition Data System for Research, or NDSR.
Okay, thank you so much for having me, and I'm so happy to be here today. Today I'll be discussing how we might integrate information about the microbiome with dietary data using vignettes from a research study that I conducted a couple of years ago now. These are my disclosures. Most relevant here is that some of the research I'll be presenting today was funded by General Mills. So I'd like to start with this picture because I really think it helps us to conceptualize the microbiome. This is a beautiful false-colored electron micrograph of the microbiome. And you can see the different members of that microbiome, the bacteria, the fungi, showing the fungal hyphae, as well as what I think is really fascinating from a dietitian's perspective, is running diagonally through the background of this image is this piece of incompletely digested plant fiber that at this magnification looks like a tree trunk. And you can see all of the bacteria are just sort of spilling over that piece of fiber, beginning to break it down. So this is a figure from Emily Leeming, who's a wonderful PhD candidate in at King's College, who works with the Twins study, the Twins UK program. And we know that the microbiome develops across the life cycle. So we know that from infancy through early toddlerhood, there's a period of dramatic change in the gut microbiome, and that sort of stabilizes as a person becomes an adult with some variation that can be attributed to differences in seasonality, time of year, location, movement between countries, exposure to antibiotics, that type of thing, medical events. And a lot of those periods of change, including the periods of rapid development during infancy, correspond with big differences and big changes that occur in the diet. So it becomes really interesting to start to think about how diet and microbiome are inherently linked. And as nutrition scientists and researchers, obviously we've understood that food we eat, potentially with some variation from our genetics, as we've had some really wonderful talks so far today discussing, influence ultimately the phenotypes that we express in our bodies that often correspond with conditions of health and disease. And for a long time, we've looked at this puzzle, if you will, without the missing piece of the contribution of the microbiome. And it's becoming more and more apparent that the microbiome is not only shaped by the food that we eat, but it also independently impacts our outcomes, and it can digest components of the food we eat to create new secondary metabolites that we previously didn't consider, which may also have an impact on our outcomes in terms of health and disease. So when I joined Dan Knight's lab at the University of Minnesota as a postdoc, I set out to answer the question, how does the microbiome change from day to day in adults? We had some good data at this point that suggests that the microbiome, if you did a big change in diet, like the switch from, say, a meat-based diet to a really plant-based vegetarian diet, you could see massive changes in microbiome conformation in the period of, say, 3 days. And we wanted to look with more finer resolution, what happens if we look on a day-to-day time scale? Can we understand more explicitly what's happening when we eat food and how it changes our microbiome? So what we did was we enrolled 34 people, and we asked them to collect a microbiome sample for us each day for 17 days, or about 2.5 weeks. And so this image is showing you the microbiomes for each of the people in our study. Along the x-axis here is day, And it's difficult to appreciate because it's quite dense. Each of these narrow bars is one person's 2.5-week timescale. So if we expand this out, we can see one individual's microbiome over this 2.5-week period with peaks and valleys corresponding with small changes in microbiome composition over that time. We can also consider the microbiome in terms of an ordination, where we try to reduce the complexity of all of the different multivariate contributors, different species in the microbiome, down to just 2 or 3 dimensions to capture the most variation possible between microbiomes. So each point on this plot represents one person's microbiome conformation. So each point is made up of all of the different members of their microbiome. And conceptually, you can see that points that are further apart in this space are more distinct and different from one another, whereas points that are right on top of each other are essentially the same. And we see that people have very different microbiome conformations, and that those conformations stay relatively the same from day to day, which corresponds with what I showed you in those relative abundance plots to begin with. So the question becomes, does diet contribute to this microbiome variation? And in order to do this, we sort of had to think outside the box. We weren't really sure of the best way to integrate dietary data with microbiome data. So this was a lot of learning by doing. We collected dietary records for the same time period. So the day before a person provided us a stool sample, they told us everything that they ate or drank during that same time period. And I won't talk about these. There are 2 individuals here who I talk about them in the paper, but they'll distract you if I don't tell you what they are. But these are people who consumed just a meal replacement beverage for the entirety of the study. And this is one of the consequences of doing citizen science, if you will. All of the people in this study knew that they were being studied for this purpose, and a subset of them were in a class who were also going to help us with analyzing the data. So 2 of the participants, unbeknownst to me, consumed a meal replacement beverage for the entirety of this study. That's all I'm going to say about them for now. But when we look at the conformation of the microbiome versus the changes in dietary intake for the same time period, you can appreciate here visually that the diet is much more variable. It's bouncing around a lot more from day to day than the microbiome that corresponds to that diet. And this is quite interesting, specifically when you think about the fact that here on the left, these microbiome points are at a really high level of resolution. So these are microbes at the species level from our metagenomic sequencing. On the right, the diet is at the least specific level of resolution that we have at food groups. So this is a very crude visualization of diet. If we tried to visualize this at the level of individual foods, which I think would be more synonymous to species, what we see is that it looks like a piece of Picasso artwork. There is so much variation from day to day in the foods that people consume that it's almost impossible to represent it. But we can look at it in terms of a network visualization. So this is a visualization that's really common in the microbiome space to understand what's happening in these complex communities. And here it's applied to dietary data. So each of the white points are the individuals in our study, and each of the colored points are the foods that a person reported eating, colored by the food group that those foods belong to. So what you can see is that in the middle of this network, which is weighted by how many people ate each food, there are foods that were reported by lots of people in the study. And so these were things like white bread, lettuce, chicken, coffee, tea, beer, things that Americans generally eat relatively frequently. And on the periphery of this network, there are foods that were only consumed by 1 or 2 individuals in the study. I told you I wouldn't talk about them again, but the individuals who consumed the meal replacement beverages are a really good representation to help you understand what's happening in this network diagram. Here, the pink dots correspond with the beverage that they consumed, and there were 4 different flavors of this meal replacement beverage, and one of these individuals consumed coffee one time, and one of these individuals consumed a slice of pizza one time. So they both tie into the network, but you can see they're very different from everyone else who clusters more in the center. And normally in nutrition science, when we try to deal with this complexity, we do so with complex error adjustment methods building off of nutrient database information that we generate from the food records that we collect. So depending on the database method you use, you get between 50 to 200 nutrients that you want to assess. And this smooths out some of the variation because we're able to get information about protein and fat and carbohydrate from each of the foods that we consume. And when we look at the nutrients reported by the participants in the study, we see that indeed it is much more stable than the intake of foods, as we would expect. So next, we took another tool from the microbiome space, that is this concept of Procrustes analysis. So we wanted to ask whether or not, if we have an ordination space of microbiome and we create one for nutrient information for the participants' diets. So here on the left, each point represents an individual's average microbiome over the study period. And on the right, each point represents the person's average nutrient intake for the study period. And then we use Procrustes analysis, which takes one set of points and holds them fixed in a multidimensional space, and another set of points, and it finds the best agreement between those two sets of points. Then it shuffles the points 1,000 times to get a representative distribution of all of the possible distances that you could create from those points. And it asks whether or not the actual pairing is better than what you'd expect. And when you look at the nutrients, we find that the microbiome doesn't pair with the nutrients. So this caused us to sort of scratch our heads, and we got to thinking about this concept of dark matter, if you will, dietary dark matter. So we're all used to looking at nutrition facts panels on the foods that we buy, and that breaks down, a selection of nutrients that is available. So it sort of represents the nutrient database, the kind of information that we can get out of a nutrient database. But on the right, you can see this visual of a strawberry, which contains many, many, many more ingredients than could ever fit on the nutrient facts panel for a strawberry. Now, some of them are in very small contributing amounts, right? They're tiny, but they are there. And there are different polyphenols, flavors, colors, compounds that we might expect from the perspective of a bacterium might be interesting and potentially tasty, if you will, if you're a bacteria. And we don't really know much about these biochemicals in our food. There's about 26,000 biochemicals in our food, which we've largely ignored, in part because the absorption of essential nutrients and minerals has been our focus for a very long time. We know a lot about the absorption of, say, 120, 200 different nutrients that may be essential or important for human health, but we don't know a lot about the other biochemicals in our food that we don't consume that make it through transit to our large intestine, where they become food for bacteria. And so from the perspective of your microbes, this is a cross-section of a mouse lumen. You can see that particles of food do arrive in the lumen relatively intact, even after digestion has taken place in the higher gastrointestinal tract, lending itself to the potential that even small components of the food could have an impact on different communities of gut bacteria. So it raises a point, how do we look at this kind of complexity when we're dealing with a lot of unknown? So how else can we look at diet? One way that we can consider looking at diet is to incorporate its highly multivariate nature, just like we do with the microbiome. So if you've ever worked with microbiome data, you know that it can be organized in terms of specific species and filled in with the amount of the species that you identified in your data. And we can do the same thing with diet. We can look in diet in terms of foods and cluster them based on food groups. So that's what we did for this study. We also pulled conceptually from the microbiome space, where phylogenetic trees arrange species into related niches. And we were able to generate not true phylogeny, but a phonetic tree of foods that we're able to then use some tools from the microbiome space to explore diet. So here is a tree of foods. This tree of foods contains things like bread next to wheat, milk next to yogurt, which allows us to statistically account for similarities between foods in terms of their food group. And we can apply a method called Unifrac to our diet data to generate dietary multivariate ordinations. And here, just like with the microbiome data, you can see that people have distinct and highly variable dietary patterns. These clouds are much larger, as we'd expect, because of the increased variation. And then we can ask whether or not food matches with the microbiome using this Procrustes analysis. And excitingly, here we've got average microbiome versus average diet in terms of food. Here we find that microbiome composition does indeed pair with food intake, which was really exciting. We did the same thing with fiber diversity. So now instead of thinking about the quantity of the food, we thought about the quantity of fiber in the food itself. And we asked whether or not grain fiber diversity paired with microbiome composition. And we found that it did. And the same thing for fruit fiber. We also found that it paired with microbiome composition. Another thing that we were able to do is to use this ordination to generate multiple orthogonal axes in a tree-based space that we can consider to be representative of dietary patterns. And these are data-driven dietary patterns. Here we found a couple of dietary patterns. We took the first 5 and we identified the main drivers of those dietary patterns and found that the dietary patterns more similar to sort of the Western high-fat diet looked like they were associated with the abundance of specific species of bacteria. And when we did what's called a constrained ordination with these dietary patterns on the microbiome data, we found that the dietary patterns explain a large amount of the variation in the microbiome and reveal a Bacteroidetes gradient, which is quite interesting. A little side note is that if we run the same approach, incorporating BMI, sex, and age, and ignoring diet, we see a large amount of the variation in microbiome also explained. And a lot of that variation overlaps with the dietary variation. So there is, as you might expect, dietary patterns correspond not only with the microbiome, but they're also driven in part by preferences that show up again in people's age and sex and BMI. So what happens at the individual level? We're talking about precision and personalized approaches. We did the same thing where we took a person's diet and their microbiome, and we asked whether or not they paired. And here we take one person's 17 days of diet, one person's 17 days of microbiome, and ask if there's agreement. And we find that for almost all of the participants, 28 of the 34, we find significant agreement between their diet and their microbiome when we looked longitudinally within a person. And when we go ahead and look at specific relationships within a person between foods and species, we find that there are a large number of significant relationships between foods and species within a person, but very few of those relationships are repeated across people. Most of the relationships were entirely personalized. And in some cases, individuals, for example, consuming dark green vegetables, that was correlated with a decrease in a specific Bacteroidetes species in one person, but an increase in that same Bacteroidetes species in a different person. So we start to understand that the microbiome community and how it interacts with food is highly personalized. So I'll leave you with one more thought, and that is that with longitudinal microbiome data, you're given the opportunity to also consider variation and variability and stability. So here you can see that some of the individuals in our study had very, very stable microbiomes, while others were much more variable, and they changed a lot every day. And what we found was that the stability of the microbiome was also associated with how many diverse different types of foods that they consumed on a daily basis on average. And some of this is starting to show up. I just saw a study in infants where they found the same thing, where more diversity of foods in this infant cohort was associated with a more stable microbiome. So we don't know what that means yet, but it's potentially very interesting. So to conclude, foods and dietary patterns help to shape the microbiome. Diet microbiome relationships are individualized. We can treat diet as multivariate data. We don't have to average out all of the difference. And we can think of it as another ohm, if you will, to integrate with multi-omics data like the microbiome, like metabolome, like the metagenomes from microbes. And then we can also consider dietary analysis in new ways. I didn't get into it, but diet looks a lot like microbiome data. So we can use the tools that we have from the microbiome space and apply it to the diet space, which allows for really awesome integration potential. And that's it.
Well, thank you, Dr. Johnson, for introducing the links between diet and the microbiome, and that the microbiome might be the missing piece of the puzzle between diet and health.